Hello, everyone. Welcome to today's live broadcast, the Catch and Release, Extraction and Purification of NGS-Grade DNA and RNA from FFPE tissue samples. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Covaris. For more information, please visit covarisinc.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem to the green Q&A button lower left. I would now like to introduce today's speakers, Guillaume Durin, PhD, Product Manager, Covaris. Dr. Guillaume Durin brings years of experience in protocol and consumables development in the field of sample preparation for molecular biology. Recently, Dr. Durin has worked on developing a DNA and RNA extraction solution for FFPE tissue by utilizing Covaris' patented adaptive focused acoustics technology and the truXTRAC purification kits. Dr. Durin?
But what type of quality is required? So you need DNA or RNA that can be amplified. Most library preparation or NGS sequencing techniques require polymerase intervention, and this has prime importance to offer reusable assays. Another requirement is to get enough DNA or RNA of high quality to make libraries for whole genome or whole exome sequencing. And when working with FFPE samples, meeting these two requirements is often a challenge. But when your extraction technique is able to do so, your library will pass QC tests and your sequencing metrics, like the coverage uniformity, will drastically improve. So today's webinar will be split in 4 parts. First, I will present the challenge when working with FFPE samples. Then I will discuss how active paraffin removal with AFA can address most of them. I will then present data showing how you can achieve NGS-grade nucleic acid extraction with AFA. And finally, I will review the full workflow developed by Covaris for nucleic acid extraction from FFPE samples. So what are FFPE samples? FFPE stands for Formalin-Fixed Paraffin-Embedded. It is estimated that there is more than 1 billion FFPE samples worldwide. And actually, it is the most common method for preserving solid tissue samples for clinical diagnostic and research. So it preserves the tissue morphology, but it has not been designed for use in molecular analysis. And so nucleic acid extraction from FFPE will enable to correlate disease state with tissue morphology with the genotype from the DNA or gene expression when working with RNA. So it's an excellent resource for new genomic insight using next-generation sequencing, but nucleic acid extraction has been highly variable and time-consuming when working from FFPE samples. So there are many challenges associated with FFPE samples. And so they are related to both the processing and the storage. FFPE samples are highly cross-linked. So indeed, formaldehyde will react with proteins, creating a 3D network that's also cross-linked to nucleic acids. Some of these cross-links can remain after the purification and affect the downstream analytical steps. It is especially true if a harsh deparaffinization treatment has degraded the proteins into peptides and before the cross-link reversal, making it much less efficient. So processing and storage conditions may also differ sample to samples and degrade nucleic acids. Standard embedding protocols will require heating, and there is no standardized embedding workflow between institutions. So we'll see large variations between samples. And also the nucleic acids in tissue block can degrade during storage, and this is especially visible with, when you're looking for RNA extraction. So what is the traditional workflow for nucleic acid extraction from FFPE samples? So first, you need to start with paraffin removal. So it's passive. You often use like some harsh organic solvent, like xylene, or something like a little milder, like mineral oils or cyclosolve. It can take from 5 to half an hour. Then you have to rehydrate the tissue. It's also passive with multiple solvent water incubation. You can then digest the tissue, usually proteinase K, sometimes other enzymes are used. Digestion time will vary greatly depending on the protocol, but it can be up to 72 hours. Then you have to cross-link, reverse the cross-link by heat, different temperature from 80 to 90 C, and up to 2 hours, depending on the protocols. And then you go to the DNA extraction purification, usually done by column purification, magnetic beads, or even some phenol chloroform protocols. So overall, it's a complex workflow, up to 3 to 4 days, with many variables. And at the end, the output has many limitations when you're looking for high quality nucleic acid. You're getting low quality because of all these steps with organic solvents, heating, you have many degradation to the DNA RNA. Protocols are complex, meaning kind of lot of variations operator to operator. Requirement for organic solvents, time consuming, up to 72 hours. And overall, poor extraction or reproducibility. One sample could yield good DNA RNA, another sample, some low quality RNA or DNA. So a critical need exists for a new method to accommodate sensitivity and quality requirement of NGS-based analysis. So truXTRAC is a different approach to nucleic acid extraction from FFPE samples. You can see on this slide the complete workflow, but in the first time, I would like to focus on the key difference of truXTRAC, the active paraffin removal and tissue rehydration with AFA-focused acoustics. And I will start by presenting quickly AFA. So AFA stands for Adaptive Focused Acoustics. It's a unique approach to paraffin removal, tissue rehydration, and homogenization from FFPE tissue samples. So first, I will present what is AFA technology, and then discuss how you can apply AFA to FFPE tissue, DNA, or RNA extraction workflow. So AFA is based on highly developed technologies which have been optimized for biomolecule processing. So one of these technology is therapeutic ultrasound, especially lithotripsy, used to break down kidney and gallstones. Another technique is diagnostic ultrasound, so low-intensity focused ultrasound used for imaging. And so combining both of these technology, we are able to completely control the cavitation and with computers, and so to have a very precise technology. So how is cavitation controlled by AFA? So I will kind of go through the different steps. So first, you have acoustic wave that will cause localized pressure fluctuations in your sample fluid. This fluctuation will cause dissolved gas to form microscopic bubbles. These bubbles will grow, oscillate, and at one point collapse. And this process will generate shear force. And AFA technology enables a precise control of the generated shear force. And so you can see at the bottom the illustration when you have at the top the pressure fluctuation, and at the bottom the variations in a radius of the bubble. So the bubble will grow and then collapse, creating this implosion, shock wave, and shear force. And so key to Covaris AFA is its ability to precisely control the generated shear force. So overall, what are the key attributes of the patented Covaris AFA? It's a non-contact delivery of controlled focus acoustic energy. And during this treatment, you have consistent sample temperature. All the energy is focused in 3 millimeter as we are working at 500 kilohertz. So the 3 millimeter wavelength, it's ideal for processing biological molecules and small samples. And so you have a geometrically focused acoustic energy. So really all the energy is delivered to the samples. You have no waste of energy around. And all the vessels have been optimized to transfer the acoustic energy. So overall, it's a highly patented technology with more than 50 granted or pending patents. And Covaris AFA can be used in a wide range of applications. So just this graph will show you some of them. And really depending, as we can finally control the cavitation and so the shear force, we can go from very low power to some immunoassay improvement, improving ELISA binding, and then increasing the power and the shear force. We can do some DNA shearing. So Covaris is the gold standard for DNA shearing for NGS. Some chromatin shearing, again, gold standard for all chip applications. And so we have also all the cell lysis applications. But today what I really want to focus is on how we can use AFA for the extraction of DNA or RNA from FFPE samples. So how can we apply AFA to FFPE samples? AFA will deliver active processing from the critical first step. So what it means? It means first we have an AFA-based deparaffinization. Paraffin is emulsified. It is an endpoint process, and it is done in an aqueous solvent. And in the meantime, as we remove the paraffin, we have an AFA-based tissue rehydration, meaning better tissue digestion by protein escape, because if your tissue is very well rehydrated, the protein escape will have access to completely digest it, and to overall improve extraction of DNA or RNA. So on the next slide, you can see some illustration of active paraffin removal with AFA-based deparaffinization. So at the bottom, you have a picture of a FFPE sample before and after emulsification. After the emulsification, you are getting a milky solution, so this white milky solution of emulsified paraffin. So the tissue inside is not visible, but still here and intact. And so when we size the small paraffin droplets, part of this emulsion, and you can see on the right, you're getting very small droplets, less than 25 micron in diameter. So it makes a very stable emulsion. And this process is done in aqueous solvent. So you're already removing all the paraffin in aqueous solvent. It's a gentle process, no heat, no organic solvent. And so it means that we don't really damage the proteins that are still cross-linked. And so when we do the cross-link reversal, we still have intact protein that we can reverse the cross-link. And so it's a much more efficient cross-link reversal at this stage. It's also an endpoint process, so completely operator independent, doesn't require any skills. It's done on Covaris instruments. And so an illustration of how efficient this process is. So you can see below a comparison of active versus passive paraffin removal. So it is a fluorescent microscopic data of different tissue sections. The level of blue is proportional to the amount of paraffin in the tissue. So on the left, you have untreated tissue, and so with plenty of paraffin. And on the right, completely at the right, you have a tissue treated with an organic solvent. And you can see you still see some blue, and so you still have some paraffin embedded in the tissue, even after the paraffin removal step. In the middle, you can see the same tissue that has been treated with Covaris AFA. And you can see that in this case, we have much less blues, meaning much less paraffin left over in the tissue. So it means that this process of active paraffin removal with Covaris AFA is both very gentle and very efficient. And so how this active paraffin removal translates into NGS-grade DNA or RNA extraction from FFPE samples? So I will then present some data showing that we tested DNA and RNA from amplificability, and we also sequenced some samples and compared genome coverage. So first data is a real-time PCR. So we choose a housekeeping gene, GAPDH, and designed 3 primer sets of various lengths. And we compare, so real-time PCR, the DNA extracted with Covaris and with another standard method. So Covaris is blue, the other method is yellow. And you can see for different amplicon lengths, from 59 base pair up to 236 base pair, that we are getting much higher amplificability and DNA yield with Covaris than with other method. So meaning that we're getting this high quality double-strand DNA that's readily available for amplification when working with the truXTRAC FFPE DNA extraction kit. So we did the same with another housekeeping gene, actin, and with even a broader set of amplicon from 109 base pair up to 600 base pair. And again, we are seeing the same type of performance, meaning that with Covaris truXTRAC, you are getting much higher amplificability, and so higher yield when you normalize to the input of FFPE material. So based on this data, we then carried out a whole genome sequencing experiment. And so you can see some results here. So how did we design the experiment? From the same tissue, we had fresh frozen and a match FFPE block. So we extracted the DNA from the fresh frozen tissue. We also extracted the DNA from the FFPE block with 2 methods, either Covaris or another standard method. Then build libraries and go to sequencing. So we choose a very low coverage, on average 10x, to really highlight difference between the extraction techniques. So as expected, the DNA extracted from fresh frozen samples, so in blue at the bottom, provide kind of a uniform coverage across the entire genome. But you can see that with DNA extracted with Covaris, you almost have the same uniform coverage across the whole genome. So slightly more variations, but still a very good and uniform coverage. But with the other technique, you can see that you have lots of variations in the coverage across the genome, so lots of peaks and valleys, meaning that you don't get this uniform coverage. So that really highlights, how the quality of the DNA that you're extracting will impact, at the end, the quality of your sequencing data, and how usable the sequencing data will be. So another feature of the truXTRAC FFPE DNA kit is the ability to control the size of the DNA fragments. So by default, we don't fragment the DNA at all during the extraction process. And so the extracted DNA will be as large as possible, and it will really depend on how it has been embedded and how it has been stored. So that's what we call option C, that's at the bottom. That's what we call extracting genomic DNA, and here you have an illustration. So with a kind of recent FFPE block, we are getting up to 8 kilobase per DNA fragments after the extraction from the FFPE sample. But we also have, and that's kind of unique to Covaris, the option to fragment the DNA after the cross-link reversal. So you're still with the emulsified paraffin, but then we can, in another AFA step, we can use the acoustic energy to fragment the DNA in a controlled way and to a size suitable for NGS applications. So it can be 200 base pair or 300 base pair, depending on type of libraries that you want to construct. So we already have these different options as part of the protocol. So by default, we don't fragment the DNA, or we can already prepare libraries for sequencing in a one-step process. So all what we saw with the DNA kit also applied with the RNA kit. So we are also getting this NGS-Grade RNA from FFPE samples. So same as for DNA, you have different ways to assess this NGS-Grade RNA, so yield, the quality, and amplificability. So I will first go on the amplificability. So what amplificability means? It's really the amount of RNA. So what amplificability means? Here you can see, first we normalize really the amount of RNA before the qPCR. And so by normalizing the amount of RNA, we already see how amplifiable each RNA molecule is. So that's the graph on the left. And so that's the number of RNA molecules after qPCR normalized by qubit as input. And so you can see that here, the RNA amplified with Covaris is already, yes, much higher, more amplifiable. But then if you take into account the initial normalization, and so the initial qubit or fluorometric dye yield, you can see that overall, when you normalize by the input of FFPE tissue, you're getting like almost 500% improvement with Covaris compared to QIAGEN. So that's really a very large difference that you can see over like different tissue type. So you can see it on kidney tissue. And so with kidney tissue, we try 2 amplicon length by real-time RT-PCR, so from both 67 base pair and 191 base pair. And in both cases, we see a large improvement in amplifiable RNA molecules with Covaris compared to other standard methods. We repeat on uterus tissue, and again, we see a higher amount of amplifiable RNA molecules with Covaris. But you can see that it's not as dramatic with uterus than with kidney or lung tissue. So that's something that really need to keep in mind, that there is large variabilities across the different FFPE tissue type. And so performance will depend both of the tissue type, but also on the storage and baking conditions. So you will see, depending of your samples, we will see a different level of improvement and usability for NGS downstream. So another way to really assess the quality of RNA molecules is a new matrix called DV200. So DV200 is a percentage of fragments above 200 nucleotides. And it's typically assessed on a bioanalyzer chip. So you can see an illustration here. So basically, everything below the 2 blue lines is above 200 nucleotides. And so with DV200, what you're looking for is a percentage of fragments between these 2 lines. So Illumina recently published a technical note evaluating DV200 as a matrix to assess RNA quality for NGS sequencing. And really just repeating the highlight. So people are usually using RIN numbers to assess RNA quality, but actually there is no correlation between RIN numbers and library yield for FFPE samples. But there is a very high correlation between DV200 values and the library yield. So their conclusion was that DV200 is a very good metric to assess the RNA quality for NGS library constructions. And with Covaris, you can see that, and see the example in the lung tissue, we are getting almost 20% improvement in the DV200 value compared to other standard methods. So the truXTRAC FFPE RNA kit also improves these quality metrics. And so when we are saying that we really get these NGS-grade DNA RNA, so that's internal data that I show you, but our customers and early access customers are seeing the same. So for example, this paper has been submitted to AACR by a group at Dana-Farber Cancer Institute in Boston. And that's just some quote from their abstract when they compare Covaris to their standard methods. So DNA yields were 6 times higher with less specimens failed extractions. DNA was more readily available for PCR amplification. And mean coverage of DNA fragments with high GC content was higher and more reproducible. And overall, library complexity was higher. So all the data that I presented, that's also what our customers are seeing. So that's still a new technique and a new product, but so we have already people publishing based on this solution. And so just recently, the Nakamura lab at the University of Chicago published in Clinical Cancer Research data of whole exome sequencing based, again, for the DNA extraction part on the truXTRAC kit. So we just focus on the paraffin removal, tissue rehydration step, but as you're familiar, an extraction workflow from FFPE sample is actually much more complex. So you have to collect the sample, from slides, or you're starting with scrolls and curves, then transfer into a tube, so remove the paraffin and then do the purification. So actually, Covaris addresses full workflow in a way that's completely scalable from 1 to 96 samples. And now I will review each different step and how we can address every challenge in these steps. So first, how we collect samples. So you have different type of samples you can get from FFPE block. And the truXTRAC protocol has been optimized for each sample type. So it's completely compatible with biopsy cores, up to 1.2 millimeter in diameter. So you just load them in a Covaris microtube and process them to remove the paraffin. It's also completely compatible with scrolls, thickness up to 25 micron. And it's also compatible with a tissue mounted on slides with a tissuePICK. And I will go into more details later. But so overall, truXTRAC has been optimized on a wide range of tissue type. So what is tissuePICK? It's really a tool that we designed to collect area of interest of an FFPE tissue slide. So it's a very safe, fast, simple process, low cost, with less operator hands-on time and variability. So how it works? So first with the tissuePICK, you determine the target of interest. Then you will capture it by sweeping the tissuePICK on the slide. And then you will release the tissuePICK into a microtube for paraffin removal. So next slide, you will see a movie. It will last 1 minute, kind of showing the use of the tissuePICK. So you won't hear any sound during this movie. It's okay. I will be back online at the end of this movie. You will also have the chance to participate in a short survey during this movie. So I'm back. And so as you can see, it's a very simple tool that you can use to collect an area of interest from FFPE section slide. So next, really, part of the workflow is what we just discussed. So once you transfer your sample, your FFPE sample, into a microtube, how you remove the paraffin with the AFA process. So we kind of went through the core technology, but I would like more, to review now more the workflow. And so we have really different way of processing these samples with AFA. It can be single tube processing, so in this type of tubes. Each tubes are individually 2D barcode. It's overall 5 units per sample to remove the paraffin. The proteinase digestion and the crossing reversal are done in the same tube. But we can also do some batch processing. So system samples are processed in SBS plate format. Each 8 strip is 2D barcode. And with a higher throughput instrument, you can process up to 96 samples in 1 hour for the paraffin removal and rehydration steps. And so this is done on a Covaris Focused-ultrasonicator, so that's really the instrument that will work and deliver the AFA acoustic energy. So each instrument that are NIST traceable for their calibration, And so you have a wide range of instruments, so scalable from a single sample to high throughput. So just going quickly through the product range, we have for single sample processing, we have the M220 with a small benchtop instrument, or the S220. Then we have the E220 EVO that can process 1/8 strip at a time. And then for higher throughput or larger batch, we have the E220 that will process so up to 96 samples, but 1 sample at a time. And the E220 that will process up to 96 samples, but 8 samples at a time. So higher throughput and enable batch process. All so truXTRAC FFPE kits are compatible with all Covaris instruments. It's just a matter of determining which instrument will fit your workflow and your level of automation required. And so the last part of a complete FFPE DNA RNA extraction workflow is really the purification. And again, with truXTRAC, you have many options. It can be either manual, so it will be columns and reagents optimized to work with FFPE lysate. So it's included in our kit. You have the columns and the reagents optimized for the purification. It's a very easy, simple protocol that you're probably all familiar with. It can also be semi or fully automated. So we are partnering with a company that really develop a completely automated solution. So it's bit-based purification. And you have different levels of automation, either semi-automated up to 12 samples at a time, or fully automated and integrated on a liquid learning robot. So again, depending on your need, many levels of customization. And finally, we also have a total nucleic acid extraction solution available. So from the same FFPE sample, we'll be able to extract both DNA and RNA in a simultaneous way, and getting, in both cases, these very high quality NGS-grade nucleic acids. And so to wrap up, what is truXTRAC FFPE nucleic acid extraction? So it's really a standardized, robust workflow for NGS-grade nucleic acid extraction from FFPE tissue. Key is a highly efficient AFA-based paraffin removal. So this is active paraffin removal, robust, optimized, in aqueous buffer, so no heat, no hazardous organic solvents. So we will preserve proteins before the crossing reversal. It is a complete tissue rehydration. So it will enable a complete protein escape digestion. So it results in maximal nucleic acid yield and, more important, quality. And so at the end, what you get is these NGS-grade nucleic acids, highly amplifiable template, unbiased coverage of the genome, and it enables really demanding applications like PCR-free whole genome sequencing. So some of our key customers are already doing this type of applications. And that's really where this is the huge importance of getting this NGS-grade DNA or RNA. So if we have one takeaway note, is that extracted nucleic acid suitability for NGS application is directly related to the deparaffinization technology. And AFA-based active paraffin removal really enables NGS-grade nucleic acid extraction. So thank you for your attention. I believe now we'll take some questions.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, why is active paraffin removal so different?
So why is it so different? That's really the way that it's done in aqueous solvent. So compared to all other techniques that are pretty harsh, either heat or different type of organic solvent, the fact that we are able to do that in aqueous buffer at room temperature means that we really preserve both the DNA and the proteins during this active paraffin removal step. And so when we go to the tissue digestion by protein escape, we have this very well rehydrate tissue matrix that will be a very good substrate for the protein escape. So we have a very efficient protein escape digestion And then, as we didn't really damage the cross-link, we have a very efficient cross-link reversal. So we don't have any like single peptide still on the DNA. So it means that we are getting a very good template then for polymerase, for polymerase for library constructions or PCR.
Great, thank you. Is it compatible with all Covaris instruments?
Yes, so it's compatible with all the Covaris instruments that we have right now on the market. And so, yes, it's compatible with all the Covaris instruments that you can use, for example, for all the library preparation for NGS. So all the instruments that you're using to fragment your DNA or your RNA, so it's compatible with this instrument and as well with all the instruments used for chip. Again, at the core of the process is the Covaris AFA technology. And so that's really what's required. This is this AFA technology to remove the paraffin, and so that's the same base brick of technology that can be used for multiple applications.
Okay, is it available or in beta access?
So yes, it's available. So the DNA kit has been available for almost 1 year. And the RNA kit has just been released, as well as a protocol for total nucleic acid extraction, and as well as a tissuePICK, so the way to collect FFPE tissue from slides is also available.
Which standard method is used for extraction Q?
That's the most common technique that used by all labs. So yeah, you can probably guess which technique it is.
Okay, and the smallest area you could take from the paraffin slide is?
So it depends really what the question is. If the question is what's the smallest that you can collect with a tissuePICK, we have like almost a 1 millimeter, 1 or 2 millimeter resolution. Then the question is, with this type of resolution, so it's like a few square millimeter tissue, will you get enough DNA or RNA out for your downstream application? It will depend, of course, on the tissue type. Do we have many nuclei or not so many nuclei? So I can't really give a black or white answer. It will depend, and also, of course, of the kind of storage condition of the slide, because when slides are stored too long, you can start to see some degradation, especially for RNA.
Okay. How long can RNA be preserved in FFPE?
Again, it will depend on many parameters, how it had been, first, how the tissue has been collected, because if it's there before to be embedded, if it's there at room temperature on a bench, you will see a lot of degradation. So if it has been like fresh frozen just after collect and then embedded in paraffin, you would get a much higher quality RNA. And then how the FFPE block is stored, it is stored as a block with paraffin layer to protect the tissue. Or is it stored at room temperature? Is it stored on slides? So many questions. If you have the choice, better to work with fresh blocks and especially blocks that you know, all the workflow and the embedding workflow, but sometimes you don't have choice. So that's so you can work with a block as long as a few years if they've been properly stored and embedded.
Okay. Is the tissuePICK affecting the sonication?
So the tissuePICK is actually quite neutral. So for those familiar with the Covaris process, we have what we call an AFA fiber into our tube to kind of control this cavitation. And actually, so we keep this AFA fiber, we just add a tissuePICK. It's just really a way to transfer the FFPE samples. It won't really affect the cavitation by itself, meaning that cavitation is still controlled by the AFA fiber that's included in all our microtubes that are part of the truXTRAC kit.
Okay. What is the minimal tissue quantity workable with AFA?
It's again the same question as working from FFPE tissue slides. It will depend on the tissue type. It will depend on the nuclei density. So yeah, we had people working with very small quantity, and also it depends. Yeah, that's many variables. Are the yield are very different depending of type of tissue that you're starting with.
Okay, next question. Can I use low material samples from laser microdissected tissue, or do I need high amounts of starting material?
It's working with both. So we have people working with low material from typically, yes, LCM, or for people working with cores. So again, it depends a lot of what you want to do downstream. If you want to do like PCR-free whole genome sequencing, you will have to start with kind of large cores to get enough DNA to do your application. If you want to do some targeted panels like Amplificability, you can work. You can work with much smaller sample input.
Okay. Are tissues that have been put on slides and covered by paraffin for a long period suitable for your technology?
In theory, yes, meaning that we already developed the tissuePICK to collect the tissue from the slide. But again, what do you mean by long period? And also, what was the storage condition? That's kind of a key parameter. If it was, some studies have shown that store in a fridge at this RNA is much better than room temperature. What was the humidity of the room? But all these parameters that will have an impact on the RNA quality. And honestly, in some cases, especially for RNA, it's very difficult. You cannot really get, regardless of the extraction technique, if the sample has not been stored properly and for too long, you can't get the quality that's required for NGS application.
All right. When in the protocol is there tissue rehydration?
So tissue rehydration is just after, and actually almost in the same time, simultaneous to the paraffin removal. So when you start with FFPE tissue in the Covaris microtube, so you have this aqueous solvent, you will deliver the acoustic energy. You have this shearing force that will remove the paraffin. And as you remove the paraffin that's hydrophobic, in the meantime, you push the aqueous buffer into the tissue, and then you have this tissue rehydration. So that's really simultaneous. Remove the paraffin and rehydrate in the meantime. You remove the hydrophobic paraffin and you push the buffer into the tissue.
Okay. Can the AFA process be applied to ADN extraction from old bones?
So can be applied to the DNA extraction from all bones. So yes, it's a kind of a different way that, and actually I can answer to that separately in an email. So yes, we have solutions based on kind of cryopulverization to work with bones. It's not working really with this kit. It's kind of a separate solution. But yeah, I can address that later.
Okay, so we have time for one more question, and it is, can we extract viral DNA with truXTRAC?
So yes, so the columns have been optimized to work with all size of DNA. So yes, we will get viral DNA if you have some yet in your FFPE tissue block. Yes.
Okay, so unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. We will respond in writing to those we did not get to today. I would also like to thank our sponsor, Covaris, for making today's educational webcast possible. Today's webcast will be available on demand for up to 6 months and indefinitely on the LabRoots webinar page. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.